Well, I'm so happy to be with you today. I want to thank Dr. Evidente for inviting me. It's my pleasure and honor to speak to this wonderful group. And thanks to Lynette Benham for organizing all of this. It's just a wonderful turnout, and welcome to everyone on Zoom as well. I'm very pleased and excited to talk to you about detecting and targeting alpha-synuclein. I feel this is just, we're just on the cusp of something really special when it comes to what we have available now for patients with Parkinson's. And we may be able to answer this question: are we on a path toward a cure for Parkinson's? Dr. Evidente helped me with this title, and he's always good at challenging me. So this is where we're going. Are we on a path toward a cure for Parkinson's? So we'll figure that out together today. All right, so we're going to talk about detecting and targeting alpha-synuclein. Right there on the left is an example of a plant where you can actually derive mucunapurians, which is a supplement many of you may have heard of that actually has some levodopa effects. And so the description was actually noted in ancient texts as long ago as 4000 BC. And then, of course, then in Western medicine by 1817, James Parkinson had described Parkinson's disease. And so this condition became known as Parkinson's disease. And then in 1912, we started to learn about the pathology of Parkinson's. So Lewy bodies, and then also the substantia nigra in terms of that cell dropout. The Lewy bodies were found to have protein inclusions. So that was around 1912. And then we have 1960 through 1990s is all basically treatments trying to manage the lack of dopamine that was apparent in patients with Parkinson's disease. Then by 1997, alpha-synuclein was noted to be associated with Parkinson's as the first genetic cause and a major constituent of Lewy bodies. So that alpha-synuclein protein was determined at that point, 1997, to be a part of those Lewy bodies that were initially described in 1912. And there on your right in the corner is that little squiggle there is an example of alpha-synuclein. We'll take a little closer look at that. And then by 2008, cell-to-cell transmission of alpha-synuclein was described and observed. And that's important in terms of how the pathology of Parkinson's progresses. And then moving forward then by 2016 or so, maybe even before, biochemical biomarkers started to be studied and examined. And so that's a lot of what our talk will be about today. In the first part of our talk, we'll talk about biomarkers and detecting alpha-synuclein. And then the second part, we'll talk about actually targeting. Go forward. So what is alpha-synuclein? And we can click forward. So that's it right there. That's the protein. So again, that squiggle at the bottom is sort of what it looks like in the body, if you will. And the A figure at the top is sort of stretching that out. What does that look like in terms of the different parts of that protein? You can kind of pick it apart. So it is a 140 amino acid protein. It is necessary for living beings, for humans. So it is actually settles usually in the nerve terminals and is responsible for neurotransmitter release. So if you don't have enough alpha-synuclein, that can be a problem as well. So this 140 amino acid protein has three different sections. There's the N-terminus, where you can actually see genetic mutations. The C-terminus is where you can see pathological mutations. And a lot of what we see in terms of biomarker detection is related to those phosphorylation sites in that C-terminus. And so sort of drilling down, looking microscopically, you can actually identify phosphorylated alpha-synuclein through those changes at that end of the protein. And that is a major component of Lewy bodies, as we mentioned. All right, and so here is an example of what it looks like at the nerve terminal. And so the top diagram there is one neuron, and it is attempting essentially to communicate to the next. The circles are what are called the synaptic vesicles. And that's what holds the neurotransmitters, right? And so the alpha-synuclein, again, that little squiggle there that you see at the top on the left, that little green, that's actually disordered protein just by itself. And then once it gets to that membrane, that vesicle, it actually attaches and becomes a little more ordered. And then it goes down to the synaptic cleft and fuses, and then neurotransmitters can be released. It may be involved in breaking some of that neurotransmitter release. That's still sort of unclear as far as the exact role, but we do feel that it is related to how neurotransmitters are released at that nerve terminal. All right. And so elevated levels of alpha-synuclein can cause disease, but lack of alpha-synuclein can cause other conditions. So the goal should not be to just get rid of all the alpha-synuclein in Parkinson's disease. We know from genetic studies and pathological studies that some conditions like autism can be associated with low levels. So precise regulation and a balance of alpha-synuclein is really the therapeutic goal. All right, and so protein misfolding, so misfolding of that specific protein is what leads to Lewy bodies and Lewy neurites. So let's take a look at that. So this kind of takes you through from the top there, again, that stretched out alpha-synuclein structure at the top, showing you the potential mutation sites and the post-translational modification sites on the right. And then B is what the structure actually looks like in the body. And then C is showing you on the left, on your left, the monomers. So that's the good type of alpha-synuclein, if you will. That's the alpha-synuclein that does its job and doesn't get in the way of anything. And in the middle, you have oligomers. So this is aggregates, essentially, of that monomer. And that can lead to problems. So the more oligomers you have that are potentially toxic, those can actually aggregate even more so and then develop into fibrils. And those fibrils can cause problems. So if you follow that down, that red, this red columns, if you will, on the right, if you look down, that neuron there on the right does not look healthy, right? So what develops are Lewy neurites that contain that misfolded alpha-synuclein and Lewy bodies. And then on the left is the healthy neuron. So everyone can see the difference between those, the healthy and the damaged neuron. And so what happens when that neuron is damaged is that it sets up an inflammatory process. So the body's trying to heal itself. And in doing so, there are inflammatory cytokines, microglia become activated, and then those cells actually potentially drop out. So those dopamine cells in the substantia nigra drop out. And that's what you're seeing there on the right. That is the midbrain. And the lack of that darker substance on the bottom is the lower levels of dopamine producing cells in that part of the brain. And then again, on the left, healthy neuron, resting microglia, they're not doing anything, they're not releasing any inflammatory cytokines, and everything's good. So that side by side really shows you what happens as far as the pathology of Parkinson's and how those dopamine cells really are damaged by misfolded alpha-synuclein and Lewy bodies and Lewy neurites. All right, so now let's get to potential biomarker candidates. So is that protein folding, does that just occur in the brain? It turns out it doesn't actually. It is quite widespread. So you can actually find that abnormal misfolded protein in several tissues. So the salivary glands, the submandibular gland, stomach, the colon, the skin. And in many cases, some of these proteins can be found years before motor symptoms. And so this is what would be called a prodromal state. So for example, folks may have constipation for years, even decades before developing motor symptoms. And patients may have smell loss, a loss of sense of smell. They may have depression or anxiety. So there's different parts of the body that reflect that pathology that is considered a prodromal state. And so bodily fluids also can show this abnormal or misfolded alpha-synuclein or phosphorylated alpha-synuclein, and the skin as well. And so we'll start with where those biomarkers can be found. Where are we with biomarkers? So just a little sort of map of where we are now. So we actually do have biomarker testing available now. So in the middle there, you see kind of at number one. So the first step in terms of testing is that the doctor and the patient discuss whether it would be helpful. And if you have typical Parkinson's that responds to levodopa, you have a tremor, you have stiffness, you have slowness, it's responded to levodopa and has sort of tracked right along with the clinical symptoms, you probably don't need this testing. It's not going to really change what we're going to do. But let's say it's very early and you're not sure, and the tremor is not very typical of Parkinson's. Maybe it's a little bit of an essential tremor possible in terms of postural tremor or action tremor. And so in those cases, you might want to do some testing to differentiate essential tremor possibilities versus Parkinson's. So the first step is really to discuss that with the doctor and to decide if it would be helpful. And then the testing happens, and then the laboratory shares the results with the patient and the physician, and then decisions are made. And so usually doctors will decide on doing a test if it means that there's going to be a change in management. So one of the changes in management, or one of the decision points, could be if you don't have major symptoms, they're very mild, maybe you want to enroll in a clinical trial. And so a lot of these trials that we're going to talk about targeting alpha-synuclein are in the very earliest stages of Parkinson's. So this is where biomarkers may come in handy. And then on the outside of that circle, we have sort of all the issues that kind of go into biomarker testing. Insurance companies have to provide coverage so that it's affordable for patients. Regulatory agencies have to approve it. The researchers have to come up with the testing. Pharmaceutical companies then can potentially develop therapies based on that biomarker testing. And so we'll start here with the first available test, which is the skin biopsy, or the SYN1 test. And so these are very small little 3 millimeter punch biopsies that are taken from the 3 different sites: the posterior cervical region, the distal thigh, the distal ankle. And what's taken is a sample of the epidermis and the dermis. And what we look for there is within the nerve structures of the dermis, you can actually see phosphorylated alpha-synuclein, which is there on the bottom and also here. So you can see that example there. The green is actually staining for the nerve, and then the red is staining for the phosphorylated alpha-synuclein. And when you see the orange, that's the co-located P-syn with the nerve fiber. So that's actually showing you the pathology in the skin. So you can actually test now for what's called a synucleinopathy. And so that's Parkinson's disease, dementia with Lewy bodies, MSA, pure autonomic failure. All of those conditions all fall under this synucleinopathy category. And REM sleep behavior disorder, it turns out, is likely one of these conditions as well. And that, again, can start maybe even 10 years before Parkinson's. So those are potentially prime candidates to where we can look at maybe disease-modifying therapies. And so can we slow or even stop the course of Parkinson's? So that's the goal. And on the right there also, it shows you the 95% sensitivity and specificity. So that test does show the protein 95% of the time, and it's specific 95% of the time. And then what you see there on the left, that diagram that just popped up, is how big that little punch biopsy is compared to a quarter, so just 3 millimeters. All right, next test. So many of you may have heard of these seeding assays. This is making a lot of headlines as far as the RT-QuIC or SAA. This was a big announcement earlier this year, and this technique actually looks at different fluids, potentially in tissues as well, where misfolded and aggregated synuclein, there we go. Aggregated synuclein from that sample, from that fluid or from that tissue, is actually then combined with recombinant alpha-synuclein. And so those seeds, essentially, so those aggregates take this substrate that is just fine, they interact, and then they misfold. So they form aggregation as well, aggregates as well. And then with that spontaneous aggregation, there's a process called quaking, where you can develop more of these seeds from that original sample, if you will. So that cycle is repeated over and over and over, and then you can detect the level of that aggregation using a fluorescent probe. And so this is how the seeding assay works, or RT-QuIC. And here on the right are all the different tissues that are being studied or looked at to see if we can actually find that misfolded alpha-synuclein. And so the CSF there is the commercially available test that made the big news and the big headlines. And so this is a spinal tap. It's called the SynTap test. So spinal fluid is taken, and in the lab, that sample actually goes through that RT-QuIC procedure that we just talked about. And the results are for sporadic Parkinson's. You can detect that about 93% of the time. And I should say in the SYN1 test for idiopathic or typical Parkinson's disease, the sensitivity turns out to be the same, about 93% as well. So the 95% is across all those synucleinopathies, 93% in the skin and the CSF really both line up. And so what's interesting here is that you may not see it in certain types of Parkinson's. Had a little less success with noting this in patients who had a normal sense of smell, also patients who had the LRRK2 mutation. So there may be something a little bit different about clinical cases where you may not see this specific biomarker. And this may be true of all of them. So we need to do more studies to see if this holds. And then also, what does that mean about those cases? Is that a different type of case where we need to do something a little bit different in terms of treatment? So this was a very exciting paper that was presented at MDS, the Movement Disorder Congress, just weeks ago in Copenhagen. And this is actually looking at potentially blood testing, so serum alpha-synuclein seeds. So using a fairly complicated technique, they actually were able to do a similar sort of seeding assay to determine if they could find this abnormal protein for different synucleinopathies as well, including Lewy body disease, MSA, and PD. And you can see there on the right the electron microscopy from the different conditions and how different they are. The filaments are a little bit different. There's multiple filaments, or there's two fibrils, and so you can potentially, or they're twisted, and so you can actually potentially then differentiate. And this is very preliminary, and of course this needs to be validated. But this is really exciting news in terms of what may lie ahead in the future in terms of potentially a blood test for Parkinson's. In terms of neuroimaging of synucleinopathies, there are possibilities. So the question is, what do we have now in terms of radiology, right? So imaging testing, what can we do now to determine if someone has Parkinson's? You've all heard of the DAT scan probably, which actually looks at dopamine transporter imaging. So that just gives you an indirect view of what's going on with dopamine, but it doesn't tell you anything really about the pathology, about synuclein. And so now there are some techniques that are being researched and not yet available commercially, but there are some research programs looking at different tracers to be able to detect synuclein in the brain. And so most of these are PET tracers, so they're small molecules or antibody radio tracers that potentially can identify generally extracellular alpha-synuclein in the case of antibodies, but in the case of the small molecule radio tracers, potentially intra and extracellular alpha-synuclein. So that's really exciting in terms of when we're looking at targets for trials, are we actually getting to the target and are we making a difference? So this potentially could be a game changer in terms of clinical trials and how we actually show that we're actually making a difference in terms of some of these clinical trials and medications. Okay, so all this about biomarkers, why are they important? Well, really, the goal is precision medicine. So the idea is that better diagnostics can give us better treatment. So in that first scenario, without personalized medicine, some people benefit and some don't. So there are patients that are all different types. So I explained to you how there's different types of cases in that one study, the Lancet study, that showed that some people tested positive, some negative, and they did have some different clinical features. Well, maybe the same medication is not going to work for all of those patients. So maybe you need different therapies. So on the right there is the idea that if you can actually diagnose folks with a specific type of Parkinson's, then you can give them the treatment that's specific for them. So you can actually study a treatment only in that population and see if it works. So potentially have some success and minimize your adverse effects. And so there may be a different treatment for different patient populations. So this is the way I think medicine in general is moving, and especially Parkinson's disease. This is sort of the next level of what we have going on for treatments for Parkinson's. All right, and so that's where we are now in terms of detection of alpha-synuclein. So how about therapeutic targeting of alpha-synuclein? Where are we? We don't have anything approved yet. I'll start off with that. So all of these are research-based clinical trials, things that are being looked at. There are a number of things that are being looked at that I won't have time to go over just in terms of the length of this talk. But I tried to choose things that were interesting and sort of phase 2, a little further along in the clinical trial pipeline. So getting closer to, are these actually going to help? And so the first step here, just walk you through this diagram in the middle in terms of how alpha-synuclein is produced. So first you have DNA, right? That's the blueprint for any protein in the body. So you've got that, and in this case, it's SNCA. That's the code essentially for synuclein. And so that then can move on to alpha-synuclein, and then aggregation can happen with aging or toxins or any triggers that may cause Parkinson's disease. And then those alpha-synuclein aggregates can be toxic, as we discussed, and turn into Lewy bodies and Lewy neurites, as you see there on the right, and then lead to neurodegeneration. Okay, so this is sort of how alpha-synuclein plays a role in terms of neurodegeneration. So the green boxes that you see above and below are potential targets. So at the top on your left, reducing alpha-synuclein expression. So that's one way to do it. So get to where you're actually creating that blueprint for the protein. You can actually reduce aggregation. So when those toxic alpha-synuclein oligomers are trying to get together and aggregate and form clumps, potentially you could reduce that aggregation. You might be able to degrade that abnormal alpha-synuclein. So there's a part of the cell called the lysosome, which is sort of the recycling or trash center of the cell. And so maybe you can make that work better to handle this toxic alpha-synuclein in terms of those misfolded alpha-synuclein p-syn molecules. And then potentially you could prevent spread. So that might mean reducing extracellular alpha-synuclein, and then you want to prevent that spread from cell to cell. And so those are the major categories of how we can target alpha-synuclein. And so the first step is called RNA silencing, or ASO or antisense oligonucleotide. So you see there on the left, again, that blueprint that I talked about in terms of how protein is made. So DNA is converted first to mRNA, and then that mRNA is converted into a protein. Well, what happens with an ASO is that it's like a little snippet of a complementary single strand of RNA that fits into that code of mRNA that's trying to make that protein, but it blocks it. It shuts it down so that protein is not made. And so there are some trials now to downregulate that RNA, and it's really alphabet soup here. There's no names for these medications yet. So BIIB094 is the first one. And this actually targets LRRK2, which we'll talk about in a little bit, which is involved in that lysosomal or trash center of the cell. And then we have BIIB101, which actually targets alpha-synuclein mRNA. And these are injected intrathecally, so it actually goes into the spinal canal, essentially. So it is fairly involved in terms of the actual delivery of the treatment. And we're in phase 1 studies for these for the most part. So that's one way that we can potentially reduce alpha-synuclein expression. And now we have translation modulators. So these are basically things that can affect the change or the conversion from RNA to protein. And so you see there on the left, the protein translation in a low iron environment. What happens is that mRNA does not go over to the ribosome because there's a special bond there at the top There's like a little complex there that happens in a low iron situation. In high iron situations, so maybe like a toxic situation, you can't have that binding of that complex. And so that mRNA goes to the ribosome and it gets turned into then from there you get a neurotoxic protein, right? So the buntanetap actually helps that complex bind. And so by doing that, you can't get into the ribosome, which means that the protein is not produced. So this is a small molecule that actually prevents this toxin from being formed. This is a phase 2 stage at this point, and safe and well tolerated, and potentially this is phase 3 now ongoing for Parkinson. So it's looking pretty exciting for this. Okay, so the next step at the top there in the middle, reducing alpha-synuclein aggregation. We'll talk about that. The first here is minzasolmin. And so this is another small molecule for Parkinson's. There's a phase 2b trial going on here. You just push that in. And so what you see here is alpha-synuclein oligomers that are bound to membranes, that this small molecule essentially gets in there and breaks up the clump. So if you've ever had tangled hair and you spray some detangling spray, this actually kind of breaks up those clumps and it makes it into the monomers that you would expect that would not be toxic. And so that's the idea behind this minzasolmin. So it's a aggregation reducer, if you will. And so that prevents the aggregation into those Lewy bodies. This is an orally available small molecule, so that's exciting because it's easier to take an oral medication than something that has to be injected. And there's a phase 2b trial going on for this. And so far, safe and tolerable. All right, and then the next thing is ANLE138B, again, more of this alphabet soup. And what you see here in the middle is that molecule that actually binds to oligomers. So your oligomers are there in the middle of those little, oh, I have a pointer. I just discovered it halfway through my talk. So you can see here these pathological oligomers. So that ANLE138B actually reduces the toxicity of these oligomers. It basically binds to it, reduces the toxicity and the toxic effects like pore formation and such that can actually damage cells. And so then you can't have, because these are no longer pathological, hopefully you can skip this step of moving on to amyloid fibrils. Now, what's important here is that this molecule actually does not bind to the fibrils. And that's important because if you just bind to the fibrils and break those up, potentially they could seed more aggregation. So what you want to do is get it at the oligomer level and reduce the toxicity of those potentially in this scenario. And so then you can have normal alpha-synuclein function. And again, some testing going on here. And so we'll see where this goes, but there are some randomized placebo-controlled trials looking at this. All right, now how about enhancing degradation of alpha-synuclein? So getting into that TRES center or the recycling center in order to get rid of those toxic aggregates. So there are what are called c-Abl kinase inhibitors that are listed there. This kind of made a splash, especially early in terms of a chemotherapeutic agent that wound up being looked at for PD and other neurodegenerative conditions. And that didn't really pan out, but there are some other candidates that are waiting in the wings, if you will. And so just walking through what the c-Abl does and how these medications work. So essentially stressors can actually cause alpha-synuclein to be produced. So this is oxidative stress, point mutations, genetic mutations, as we talked about early in terms of what happens with that alpha-synuclein. So toxins, inflammation. So then when these aggregates start forming, the c-Abl, it's actually sort of like a sentinel or a watch. It's like a guard, essentially. It's looking out for these abnormal aggregates inside the cell. But the problem is that this guard doesn't do a great job because it makes things worse. It actually takes those aggregates, modifies them further, and it makes them more toxic. So what you want to do is actually inhibit that c-Abl, so the c-Abl kinase. And so that inhibition then can prevent moving on towards neurodegeneration by reducing that toxic aggregation. Okay, and then now LRRK2 inhibitors, another alphabet soup medication here, BIIB122 or DNL151. And so what you see here are 3 different examples of different cells and what that lysosome does with LRRK2. Healthy cell, you're all in balance, everything's good, alpha-synuclein levels are controlled. With the diseased cell, you have an elevated LRRK2 actually, which causes dysfunction of the lysosomes. And so they can't handle these aggregates of alpha-synuclein. So they sort of sit around and they cause further damage. And so if you can inhibit that LRRK2, reduce the level of that, then that lysosome can work properly and manage the aggregated, potentially aggregated or toxic, just beginning oligomers, it can handle that better. So this is a phase 2 study. It's called the LUMA study that is recruiting in early Parkinson's. And then we have Ambroxol, which is actually a cough syrup that's available that increases glucocerebrosidase. It's a phase 2 study looking at PD and GBA, a specific mutation in Parkinson's disease. And basically what this does, what you're seeing on the right here, nucleus, the rough endoplasmic reticulum, the Golgi apparatus, basically these are ways that that GCase or glucocerebrosidase actually helps. The Ambroxol actually is a chaperone. And so the little blue dots are GCase. And so if you can get a chaperone in there to get it through the Golgi into that lysosome, then you can potentially handle this insoluble alpha-synuclein or pathological alpha-synuclein and reduce the potential for Lewy bodies. So that's the idea behind increasing GCase, is to basically chaperone the Ambroxol, is to chaperone GCase through, get it to the lysosomes where you can again handle the garbage in the cells, if you will. So far safe, well tolerated, good CSF penetration and studies so far. Phase 3 will start soon for PD and patients with the GBA mutation. And there are a lot of phase 2s going on as well for this in GBA as well as PDD and other conditions. And as I said, it is a cough syrup that's used to treat respiratory illness. I think it's more available in Europe, I want to say, not here in Asia. Okay, got it. Yeah. So that's the idea behind Ambroxol. So we'll stay tuned for that one. All right, so how can we prevent spread of alpha-synuclein? So one way is to actually reduce extracellular alpha-synuclein. So this has been looked at already with monoclonal antibody studies. So prasinezumab and simpanimab are monoclonal antibodies against alpha-synuclein. This study here, this molecule has been abandoned. It didn't really go anywhere. This antibody, this IgG monoclonal antibody, did not meet the endpoints for the original study in early PD to slow progression, but there may be some signals that it could prove to be helpful. And then we have, so that's really passive immunity. So you're providing this monoclonal antibody to take care of this abnormal alpha-synuclein. What about if you can boost your own immune system against alpha-synuclein? Well, that's what's really exciting about this UB312. So let's take a look at those in a little detail. So this is the New England Journal article that looked at prasinezumab. And what you see here is placebo. And then you also see the low dose and the high dose here, that towards the end of this study, there was a trend towards stabilizing the change from baseline in terms of the motor testing. So even though it didn't meet the full endpoints, what was expected, there's a signal that's strong enough that there will be sort of an extension of this trial. So they're not abandoning this just yet. But so the jury is still out on this one, essentially. But in terms of the active immunity, the UB312, this is really exciting. So you can actually potentially induce antibodies against abnormal alpha-synuclein. And it's based on this synthetic peptide vaccine technology. It has been studied in a placebo double-blind controlled study. The antibodies do cross the blood-brain barrier, which is necessary for managing alpha-synuclein. And there's potential target engagement as well, which is exciting. All right, so all of that to say, are we now on a path toward a cure? So there's multiple paths here, right? That's quite a highway system, probably LA maybe. So here is as of January 2023, and it's this is expanded beyond that since then. We're already into September. But what this is basically all the candidates for medications for Parkinson's. Now at the top there, you see disease modifying therapies. So DMT, disease modifying. What does that mean? That actually means, can we slow the progression of Parkinson's? So all of those molecules, treatments at the top there are actually looking at maybe, can you slow down Parkinson's? Can you modify the disease? So that's all at the top. And then at the bottom here are STs or symptomatic treatments. So these are treatments for patients who already have symptoms. What can we do for patients here? Are we able to manage symptoms better? So phase 1, phase 2, phase 3 are the different phases that we look at in the clinical trial pipeline. As far as first we have to look at safety, then we have to look our dosing, then we have to look at safety, both together. Then we have to figure out, okay, maybe there's some benefit here, but we still need to figure out safety and efficacy and adverse effects. And then phase 3 is where you're really doing your best to study this in a specific condition to see if it's efficacious. So these are the phase 3 studies sort of in this target here. And so there's symptomatic, there's disease modifying. The alpha-synuclein based therapies are in yellow or highlighted. Actually, some of the highlights went away. Sorry about that. But it's basically the orange here. So all of this here, UB312s, some of these are repurposed drugs, Buntanetap that we talked about. So those are the only ones that are targeting alpha-synuclein. So we have a wide range of different potential therapies and different potential targets beyond alpha-synuclein. I think this is one really exciting thing that we can look at and target. But I think it's going to wind up being multiple. I think we're going to wind up with a multiple, sort of a tiered strategy. So alpha-synuclein may be part of it. There may be cell-based therapies where we're actually replacing cells. Maybe we need the active immunity to make sure that those cells don't develop that toxic alpha-synuclein and degenerate themselves. So I think we're probably going to be looking at a tiered strategy. Oh, there's the highlights for you. How about that? Okay. All right. There we go. Those are all the ones that are targeting alpha-synuclein. So here's my take. Are we on a path to a cure? So you see here we have A, B, C, D, E, F. These are multiple paths. If you were a mountaineer, these are all the possible paths to get up that mountain. I am not a mountaineer. I would not dare to do this, but it's nice to know that there's multiple paths. And that's what I think is going on here with Parkinson's. I think that alpha-synuclein is one of many. I think that we have many potential paths for a cure. I think that the cure will probably be multi-pronged or tiered. And I think we can get there. I think there's a lot of exciting possibilities. There's machine learning to figure out, what's actually happening with these studies, and what are the molecules that have the best chance of working. So I think we're going to potentially get there. So I would say, I think there's potential for multiple paths to a cure. That's my answer. What's yours? All right. Thank you so much for your attendance and for your interest.